Hi everyone, these are anatomy and physiology videos for healthcare students. In this video, we'll be discussing enzymes. So enzymes are often introduced during anatomy and physiology when we discuss organic molecules. You may have been discussing proteins when these are introduced, but you may not hear about them again until you get to a unit on digestive system and their enzymes. So we'll talk about enzymes and how they operate here. So enzymes are considered to be a type of organic molecule called a protein. So they follow the rules of the proteins. For instance, heat can denature an enzyme, so can temperature, so can a high salt concentration. So just like proteins have optimal conditions in which they operate, so do enzymes. Enzymes are a special group of proteins. They're considered to be catalysts because it is their job to help speed up and run a reaction more efficiently. So we often say they're going to speed up the reaction. However, they are not used up by the reaction. They're available to run reaction upon reaction without being consumed by that reaction. We need to understand a few key terms before we talk about enzyme reactions. Reactants and products are often discussed during enzyme reactions. Enzymes are going to often help to assist with the transformation of reactants into products. So the reactants are the chemicals that enter into the chemical reaction. They are the ones changed by that reaction. The products, however, are the ones that are resulting from the chemical reaction. So an example we could use would be the ingredients to bake a cake. Your ingredients will be entering the chemical reaction. You'll mix them together. You'll apply heat by placing them in the oven. Wait about an hour and you'll pull out your product, and that's going to be a cake. So that results from the chemical reaction. This is a typical enzyme reaction. You can see on the left-hand side, we are looking at the enzyme plus the substrate. And on the right-hand side, the arrows point toward the enzyme and the products. So the reactants here are the enzyme and the substrate. The products are the enzyme plus those two little products shown on the right-hand side of the equation. Let the arrows guide you. They are always pointing toward your products. So in biology, we often use those arrows pointing toward the right. But if you take chemistry classes where you see those arrows pointing toward the left, that's because that's the direction the reaction will run. But, for us, that reaction is running, in this case, toward the right. It is resulting in the enzyme in those two products. You will often hear that enzymes help to lower the activation energy. Activation energy is the energy needed to start the chemical reaction. It's the energy needed to make the reaction run. You may have heard of catalytic converters in a vehicle. That helps the engine to start. In other words, it helps the reaction to turn over. So since enzymes are catalyst, like catalytic, that same term there, that's a great way to remember that they're helping us to speed up the chemical reaction. One way they do that is by lowering the amount of energy needed to start the reaction. So to understand how activation energy works, let's imagine that your task is to push this 400-pound ball up and over that really tall hill. It's all you. There's no enzyme to help you. So it's completely your task alone to huff and puff, push that ball up over the hill. So it requires a great deal of activation energy if you're going to accomplish that task. So how does an enzyme help us? Let's cut the hill in half. That's how the enzyme helps. It lowers the amount of energy required to start the chemical reaction. In other words, it lowers the activation energy required to run the reaction. Now, if you're deciding that, okay, my job is to push this 400-pound ball up and over the hill, you're going to do some huffing and puffing, but not nearly as you would in the previous image. So you still have to have some activation energy to initiate the reaction, but not as much. So in our bodies, when we break down things like proteins, we do require an input of energy to help break down those proteins. Without enzymes, we would actually heat up. As it is, we do end up spending energy to break down and digest proteins in the foods that we eat. So the bottom line is enzymes help lower the amount of energy it takes to start a chemical reaction. We say enzymes lower that activation energy. So let's talk about enzymes and the types of chemicals they work on. The chemicals they work on are known as substrates. Enzymes are built specifically for particular substrates. They are not a one-size-fits-all kind of reactant. It is not a multi-tool. It is simply a type of protein that is built to work on only one fit. And we say that they are substrate specific for that reason. In this image, let's take a look at the enzyme and how it interacts with its substrate. In this image, that blue square is your substrate. When the enzyme comes together with its substrate, it fits at an active site. And we'll say that the substrate has a lock and key fit with that active site of the enzyme. So you'll notice these two look like they're made to fit each other. And usually that's the case, or maybe we need a little help and we can do an induced fit. But for the most part, it looks like the substrate and enzyme were made to fit together at that active site. And that's where the chemical reaction will take place, is along that active site. So here you've got a substrate. It's green in this picture. And you may say, well, it doesn't really look like it's a good fit for that enzyme's active site. So this particular enzyme and substrate, they are not a good fit. There is not going to be enough of an induced fit to make these work at that active site. So this is not an enzyme that can work on this particular substrate. We need a better fit at the active site for them to work together. Let's come back to this image again. So we've looked at this one already to discuss reactants and products. So now let's look at it from the perspective of what are we doing to that substrate. So we see on the left side where the reactants are located, the enzyme acts on the substrate, and the end result is to produce a pair of products. It looks like we're taking a polymer, perhaps two pieces, breaking it apart, and we're releasing that into two monomers. So this is a catabolic reaction. Catabolic means it is a breakdown reaction. So here, the enzyme has helped us catabolize or break down that substrate into two products. We can also say the enzyme has helped us catalyze, which means it has helped us speed up this chemical reaction. Let's take a look at this one. So in this reaction, you'll notice the enzyme fits together with two substrates. Here, they are labeled as S1 and S2. And through the course of this chemical reaction, by the time you reach letter D, you have a product that has been assembled from two individual components. In other words, monomers came together to create a polymer. So this is classic an anabolic reaction or anabolism. We have monomers joining to create a polymer. And enzyme help to catalyze that reaction by bringing those two substrates together. It helped to speed up this chemical reaction. So left by themselves, it may take a lot longer and a whole lot more input of energy for those substrates to become joined together and chemically bonded. So yeah, look for those substrates. They are individuals in the reactants. And then the product, you can see they have joined together. That's how we confirm this is an anabolic reaction. Here's an opportunity for you to label the enzyme reaction. You may want to pause the video here. Take a moment, figure out those four components. All right, so enzyme in this picture has the active site on it. That active site is specifically where your enzyme and substrate will come together. Now, the substrate is the chemical upon which the enzyme will act. And you'll notice this time we're creating two products as a result of the enzyme reaction. So as a bonus, is this a catabolic or an anabolic reaction? Well, it is breaking apart that substrate into two products. It is a catabolic or a breakdown type of reaction. So we can say the enzyme catalyzes or speeds up this catabolic reaction, which is a breakdown reaction. Thanks for watching my video. If you have any questions, please leave them in the comments for me. And if you like this video, please follow my channel.